Let me just pull up my slides. Okay, can everyone see my slides?
Yes.
Great. Great. Yeah. So my name is Kevin Duclos. I'm a postdoc at the University of Oregon, and I'll be talking today about unsteady sinking behavior in diatoms. And so diatoms are unicellular phytoplankton. They don't have the ability to swim. So we might not really think about behavior when we think about diatoms, but one of my goals of this talk is to show you that diatom sinking is a very complex phenomenon that well controlled, and it might indeed be fair to talk about it as a behavior. So before I get started, I'd just like to acknowledge my collaborators on this work: Brad Gemmel at the University of South Florida, Lee Karpos at the University of Maine, and Glenn Wheeler at the Marine Biological Association in the UK. And we actually met Glenn at the last microscale meeting in Whistler. So it's appropriate that I'm presenting this work here. And some of the earlier work was done in collaboration with Tracy Villarreal at UT Austin. And our funding source was the National Science Foundation. So I'll just get started a little bit with a background on diatoms and diatom sinking, why we should care about it, and what affects it. And then I'll talk more specifically about the effects of light and nutrients on diatom sinking. And finally, I'll try to answer the question of whether unsteady sinking increases nutrient flux to a sinking diatom cell. So a little bit of background about diatoms. Diatoms are a large and diverse group of marine phytoplankton. They're found throughout the world's oceans, also in freshwater. And probably their most conspicuous feature is their frustule. So this is a silicate containing shell that surrounds the cell, and as you can see from these drawings by Ernst Haeckel, they take a variety of forms, and they can be quite beautiful. But for the purpose of this talk, the reason they're important is because they contain silicate, and so they're more dense than seawater, and they cause diatoms to sink. So why is diatom sinking important? Well, from a global perspective, sinking is an important contributor to flux of nutrients from nutrients and carbon from the upper ocean. So export of diatoms from the upper ocean to depth has been found to contribute up to 40% of particulate organic carbon export. So this is quite an important component of the biological pump, which removes carbon from the upper ocean to depth where it can no longer interact with the atmosphere. From the perspective of the diatoms themselves, from diatom biology, diatoms require both light and nutrients. And unfortunately for diatoms, light and nutrients are spatially segregated. So irradiance decreases with distance from the surface, whereas nutrients are found in higher concentrations at depth, generally because phytoplankton in the upper part of the water column quickly use up available nutrients. So a diatom's depth is very important in determining its access to light and nutrients. And it's the combination of sinking and advection that determines a diatom's depth in the water column. Sinking will obviously cause a diatom to go deeper in the water column, and advection is just the movement, the background movement of the water that can move the diatom in any direction. In addition to its importance in maintaining the position in the water column, sinking is important because it can increase nutrient uptake rates. So it's been found that for some cells, sinking can greatly increase nutrient fluxes. Diatoms are diffusion limited, so they can only take up nutrients as quickly as they diffuse in. But by sinking, diatoms can actually thin their diffusion boundary layer and increase nutrient uptake rates. And I'll come back to that later on in the talk. So for all these reasons, diatom sinking is an important part of many ecosystem and nutrient cycling models. But this term is not very well constrained. Often models will just use a single value for a diatom sinking speed, or maybe they'll pick 2 or 3 size classes and pick a sinking speed for each. But as I'm going to show you, it's quite a variable phenomenon, and it varies over multiple timescales. So diatoms, maybe not surprisingly, are actually able to regulate their buoyancy. And they do so in response to various factors, abiotic factors such as temperature, irradiance, salinity, and turbulence. And also biotic factors like nutrient depletion. So not only the concentration of nutrients in the water is important, but also sort of the history of exposure to nutrients. And also reproductive status. So diatoms have different sinking behaviors that they undergo depending on where they are in their reproductive process. So it's not entirely understood how diatoms are able to regulate their buoyancy. There are a few different mechanisms, and it's probably a combination of a few of these. It's known that diatoms can exchange heavy and light ions with seawater. So that's one way of regulating density. Another is production of organic osmolytes. And it's also been shown mathematically that active water pumping could be a way of regulating buoyancy, although as far as I know, it's not actually been experimentally confirmed. And so a lot of our knowledge, probably most of our knowledge of sinking in phytoplankton comes from settling column experiments, or SETCOL. And SETCOL was formalized by Bien Feng in 1981. And the method is fairly simple. The phytoplankton of interest are put in a water column with a known height, and then the amount of diatoms or other phytoplankton that reach the bottom is quantified over time, and then based on the fraction of phytoplankton that have sank over a given period of time, it's possible to calculate a sinking speed. And this has really given us a lot of knowledge on sinking. A lot of the papers that I cited a minute ago are based on this method, but there's a limitation to this method also, and that's that it only provides one number. So it gives a mean sinking speed for an entire population for whatever you put in that column. So more recently, we and others have used a video-based method, and the advantage of this method is that it actually allows us to track individual cells in real time. So this is sort of a schematic of the setup that we use. Most of the work I'm going to be showing use this setup. We have a filming vessel, and this is quite large with respect to the diatoms. We use one 10 by 3.5 by 30 centimeters, and we introduce the diatoms in close to the center of the top of this filming vessel, and we watch as they sink through near the center of the tank. And the advantage here is that we're far from any walls, so we minimize wall effects. And we also use a salinity gradient in this filming vessel. So we have high salinity at the bottom and low salinity at the top, and we have a very gradual salinity gradient. The purpose of this is to minimize any ambient motion of the fluid. And this is quite important because diatom sinking speeds are very slow. So any ambient motion quickly overwhelm the sinking speeds.
I'll tell you something, if I change my mind, I'll let you know. Right now.
Excuse me, I think somebody has their mic turned on. Okay, so this is these are the cells that we're going to be looking at. This is Coscinodiscus, and we'll look at a couple of different species of Coscinodiscus, wailesii and C. radiatus. These are both large centric diatoms. C. wailesii is a little bit larger than C. radiatus, but they're both sort of unusual in their size. So the first thing I'll show you is sort of the limitation of using just one number to look at sinking speed. So this is the results from an experiment with Coscinodiscus wailesii. These are 143 cells from the same culture, same exact treatment. And we can see there is a mean sinking speed, which is 0.2 millimeters per second, but there's also quite a range. So we have cells that are sinking as slowly as 0.01 millimeters per second and cells that are sinking as quickly as 0.68 millimeters per second. So it's probably not really fair to use just one mean sinking speed. And as you'll see, this is a video from one of our experiments. Not only are the cells not sinking at the same speed, individual cells are actually changing their sinking speed and doing so quite quickly. So this video is sped up a little bit. This is we're playing back at about 3 times speed. But what you're seeing here is an unsteady sinking behavior where diatoms are alternating between very slow sinking periods and then faster sinking of, in some cases, up to almost a millimeter per second. And we call this start-stop behavior because there's a clear, it's sort of these 2 clear phases of slow and fast sinking. And I just found these videos mesmerizing. I think this is such a fascinating behavior to watch. And it's sort of motivated our work on this phenomenon. And here's just another way of looking at the same thing. So this is a frame stack where we have evenly spaced frames. And so you can see nicely where the cells are bunched up. That's periods of slow sinking, whereas when they're farther apart, that's faster sinking. And so this was first documented in a paper by Gemmell et al. In 2016. So what we're looking at here is these are sinking speed plots for individual cells. In black is a cell that was depleted of nutrients for 6 days. So this is has the strongest unsteady sinking behavior. And again, we see these periods of fast and slow sinking and a sort of oscillation between the two. We can compare this to the heat-killed cells in green. So that just demonstrates that this is in fact something that the cells are actively doing. And the other thing we see here is that there's some effect of nutrients. So the nutrient replete cells here, they're just straight out of the media, are shown in red. They have some degree of unsteady sinking, but as we deplete the nutrients, first for 2 days, shown in blue, and then for 6 days shown in black, we get more and more unsteady sinking and faster sinking as well. And so this suggests that there's some, that nutrients are important for this behavior. And if we go back to sort of what I mentioned at the beginning, sinking can increase nutrient uptake rates. So it sort of makes sense that there may be some role of nutrients in this behavior. We don't know a whole lot about the mechanism for unsteady sinking, but we do know that actin and myosin seem to be important. So if we expose the cells to a myosin ATPase inhibitor or an actin inhibitor, we see in these dotted lines here, we see that the unsteady sinking behavior almost completely disappears. So to understand this behavior in a little bit more detail, we looked at the effects of light and nutrients in a series of experiments. So first, we were interested in how unsteady sinking behavior responds to varying nutrient and light conditions. We saw from before that nutrients seem to be important. Now we're adding the effects of light. So we exposed wailesii cells to either high or low nutrient concentrations for 12 hours under either light or dark conditions. So again, here's a plot of sinking speed for an individual cell over time. And we see the slow sinking punctuated by these fast sinking periods. And this is a nutrient depleted light condition. If we compare that to the nutrient replete light condition, so these are our cells that were in media for the duration of the experiment. These have lower mean sinking speed and less unsteady sinking, as we saw before. Now, if we look at the nutrient depleted dark condition, we see that unsteady sinking is almost completely, the cells are almost completely steadily sinking, and that the mean sinking speed is much less. And if we can overlay these just to compare them a little bit more easily. And those were results for individual cells. And if we look at the summary of all the cells, each treatment had at least 100 cells, we can see the overall relationship. So as we saw before, the nutrient depleted cells sank much more quickly and more unsteadily. You can also see that there is a greater range of sinking speeds. And so this sort of fits with the idea that this behavior helps diatoms to take up nutrients. As they're depleted of nutrients, they may sink faster in order to replenish their nutrient supplies. And then if we compare with the nutrient depleted dark condition, we think that this may be a mechanism to prevent the cells from being completely removed from the euphotic zone. So when they encounter the dark, slowing their sinking speed will prevent them from being exported too quickly. So to sum up, the diatoms sank the fastest and most unsteadily following 12 hours exposure to the nutrient depleted light conditions. And faster sinking by the nutrient depleted cells may be a way of increasing nutrient uptake rates, whereas slower sinking in the dark may help diatoms avoid being exported. Next, we looked at whether unsteady sinking behavior responds differently to different nutrients. So we exposed diatoms to nutrient media that was depleted of just one nutrient, nitrate, silicate, or phosphate for 24 hours. And then we added a spike of the missing nutrient at time 0. So here's the result for nitrate. At time -24 hours, we put the diatoms in media that was repleted in all nutrients except nitrate. And we can see that over 24 hours, the mean sinking speed gradually decreased. And then at time 0, we added nitrate. And then we can see in the next 2 hours, the mean sinking speed greatly increased and unsteady sinking increased as well. We see a similar result for a silicate. After that addition of that nutrient spike, of the silicate spike at time 0, we see a great increase in mean sinking speed. Here we don't see any significant change in unsteady sinking. And then if we contrast this with phosphate, we see that actually the spike of the nutrient has caused a decrease in the mean sinking speed and a decrease in unsteady sinking. So to sum up, we saw that exposure to nitrate or silicate, after 2 hours, the mean sinking speeds increased and then declined over the next 22 hours. But we saw the opposite effect for phosphate. And we're not entirely sure what's responsible for this difference, but we have a few possible explanations. One is that maybe diatoms just don't use sinking to increase their phosphate uptake rates. This one seems less likely. Phosphate is quite an important nutrient for diatoms, so it would be a bit unusual if they are not taking advantage of the nutrient spike. Another possibility is that the cells were not fully depleted of phosphate. So what we're seeing is sort of just a response to a nutrient replete condition over time. And then another possibility is that there was a sinking speed increase, but we missed it because it was fell within that 2-hour period. And as I'll show you, diatoms actually can respond quite quickly to changes in nutrient conditions. So next, we saw that the diatoms could respond within 2 hours to a change in nutrient concentration, but we wanted to see if they could respond even more quickly. So we did an experiment using a nutricline where we had low nutrients on the top and high nutrients on the bottom. And we tracked cells with 2 cameras simultaneously, one above and one below the nutricline. These cameras were about 10 centimeters apart. So here we can see the experiment. We just added fluorescein in with the nutrients. So you can see those two, that above and below the nutricline. And this also shows us just that the salinity gradient is effective. We see very little mixing between the top and the bottom. And just to clarify, in this experiment, we don't have a steep salinity gradient. It's just a steep gradient in nutrients. So we still have our same gradual salinity gradient. So what you see here is as the cells move from low nutrients to high nutrients over that 10 centimeters, we see that the mean sinking speed increased by about 8 times, and the standard deviation of sinking speed, the unsteadiness, also increased significantly. So these diatoms are responding quite quickly to changes in nutrients, and this was somewhat surprising. Before it was only known that diatoms could respond to nutrient changes, but most of these experiments happened over several hours to days. But this sort of makes sense that diatoms have this ability. Nutrients are not homogeneous in the environment. They're quite patchily distributed, and so it would be quite an advantage for a diatom to be able to respond to patchy nutrients in over scales of centimeters, where these patches are actually the scales that these patches actually occur at. And we can see from all of these results that the effects of nutrients on mean sinking speed are actually a bit complex. There's not just one sort of result. If we look at shorter time scales between 30 minutes and 2 hours, we saw that sinking speeds increased with exposure to nutrients. So that exposure to that spike caused an increase in sinking speed. Whereas if we look over longer periods, 12 to 24 hours, sinking speed decreased as the, so this is sort of gets into that effect of nutrient history, where diatoms that are replete in nutrients, maybe they've already replenished their stores, they don't need more nutrients, so then they slow their sinking speed. So now I'm going to try to answer the question of whether unsteady sinking increases nutrient flux. So as I mentioned briefly at the beginning of the talk, sinking can increase nutrient flux to a cell. This is a plot of a perfect sink model where we have on the left side of the x-axis we have a cell, and as we move along that x-axis, we're moving farther from the cell, and on the y-axis we're looking at the nutrient concentration. So this just shows us how the nutrient concentration changes as we get farther from that cell. And so, as I mentioned, diatoms and other small cells are diffusion limited. So there's what's known as a diffusion boundary layer. This is usually defined as a region where the concentration equals 0.9 times the concentration far from the cell, C infinity. And this means that diatoms are only able to take up nutrients as quickly as they diffuse into the cell. So for a nitrate or phosphate molecule to reach a 10 micron radius cell, that takes about 8 seconds. And this equation is a nutrient flux to a cell using this model. And so you see that it depends on the diffusion coefficient and the concentration at C infinity and the concentration at the cell surface, which we set to 0, and then the size of the cell, the radius of the cell. And if we set the C infinity to be 1, we can simplify this to say that the flux just depends on the diffusion coefficient and the radius of the cell. So here's the same thing, but just looking at it surrounding a spherical cell, we can see the concentration surrounding a cell. This is actually reversed here. We're looking at diffusion out from a cell, but the equation's the same. Just the sign has changed. Now, if we look at a sinking cell, we can see that these lines of concentration, they get sort of bunched up around the cell, and we have steeper gradients in nutrients. And these steeper nutrient gradients, this thinning of the diffusion boundary layer actually causes increases in nutrient flux to the cell. And so there's a dimensionless number that quantifies this increase. It's known as the Sherwood number, and it's simply the ratio of fluxes to a steadily sinking cell and a stationary cell. And the Sherwood number depends just on the Peclet number, which is a ratio of advective to diffusive fluxes using this equation. So we can, just from the Peclet number, we can calculate what the nutrient advantage of a sinking cell is. And so the Peclet number depends on the sinking speed, a cell radius, and the diffusion coefficient. So because the Sherwood number, it depends just on the Peclet number, and the diffusion coefficient is fixed, there are sort of two ways for a cell to increase its nutrient uptake. It can increase its size, or it can increase its sinking speed. But there are disadvantages to both of these. So an increase in size also decreases the surface area to volume ratio, and increasing sinking speed will remove the diatom from the photic zone more quickly. So we were interested in whether unsteady sinking might sort of solve this problem in some ways. So the question that we're interested in is whether an unsteady sinking cell can increase its nutrient flux as compared to a cell that's sinking steadily at the same mean sinking speed. So in this way, the cell will not be exported more quickly by sinking at a faster mean sinking speed, but it will still gain the advantage of increased nutrient flux. So kind of getting the best of both worlds. And in order to quantify this possible effect, we define another dimensionless number, which we call the enhancement number. If anybody has a better name, I'd be happy to hear it. And this is the flux of nutrients to a cell sinking unsteadily to one sinking steadily at the same mean speed. So the Sherwood number depends just on the Peclet number, but this enhancement number is a bit more complicated. It also depends on the cell radius and diffusion coefficient, which we saw in the Peclet number. But there are two sinking speeds. So we have a V fast and a V slow, which is sort of going back to those plots that we saw where sinking speeds were alternating between fast and slow sinking. And we have the periods of each of these, so a fast sinking and a slow sinking period. And so to look at how these different factors affect this enhancement number, we use a CFD model of a sinking diatom. So we have a spherical cell here, and this is an axisymmetric model. So we're just seeing half of the cell here. And we use a moving mesh, and that allows us to account for acceleration from the unsteady sinking term. And we use a scalar transport model to look at the flux of nutrients. So what we're looking at here is nutrient concentration surrounding a sinking cell with high nutrients far from the cell, and then this depleted region surrounding the cell, and you can see how it's how the wake of the sinking cell is sort of stretching this region. And so now if we look at the results from this model, we're looking at nutrient flux over time to that cell. In black, we see the sinking speed. So this is a sort of a simplified model of that stop-start behavior. We have the slow sinking and the fast sinking periods. And then as the sinking speed increases, we see that the nutrient flux increases as well. And we can compare this unsteady flux to the steady flux to a cell sinking at the same mean speed. And what's important here is the integral of each of these. So if the integrated flux to an unsteady cell is greater than the integrated flux to a steadily sinking cell, then we have enhancement. So now if we go back to that perfect sink model, we remember that the nutrient concentration at the cell surface was set to 0. And then we have a concentration of 1 far from the cell. And the flux was calculated by dividing the diffusion coefficient by the cell radius. But this is not quite a realistic model. Diatoms are not going to be taking up all of the nutrients available to them, so there's some limitation to their uptake rate. So we use a nutrient uptake model, sort of a simplified one, somewhat analogous to enzyme kinetics models. we defined a nutrient uptake parameter, which we call Cmax. So in this case, the nutrient concentration at the cell surface, C0, we're not setting that. We we let that vary, and we calculate the flux by multiplying this Cmax, this nutrient uptake parameter, by the concentration at the cell surface. And so here are the effects, here are the results. So if we look at that nutrient uptake parameter, if we look at the right side of this plot, we see something closer to that perfect sink model where the diatoms are taking up most of the nutrients available to them. But then if we go to the left side of this plot and we decrease that Cmax, then we start to see enhancement. We see that the diatoms are actually increase their uptake rate by about 13%. And we can also look at other parameters. So we can look at the fast sinking speed. And here we see that as the fast sinking speed increases, that nutrient uptake, that enhancement number increases as well. To conclude this section, unsteady sinking can increase nutrient flux under some conditions, not in the case of a perfect sink model, but we do see greater enhancement in the cases where the uptake parameter is lower and where the fast sinking speed numbers are higher. And we're still working on this model, testing other parameters. So this is a bit preliminary. So most of the work we've done was with the Coscinodiscus, but we're interested in whether other cells could also exhibit unsteady sinking. And sort of just based on the Sherwood number, we would think that the cells that benefit most from unsteady sinking would probably be large and fast sinking cells, because these are the cells that benefit most from steady sinking. So we were a bit surprised to find that these cells, which are much smaller, diatom bright wailesii, are actually do the same thing. And so here's a video of a chain of 2 diatom cells, and we're seeing unsteady sinking. But another thing that's kind of interesting about this video is just one of the 2 cells in the chain is changing its sinking speed. So we're seeing this kind of interesting flip behavior. So we're interested in how widespread unsteady sinking is. Just based on our preliminary results, it seems to be much more widespread than we had expected. And we're also looking at the cellular mechanism. So we have some experiments planned with Glenn Wheeler to try to get at those questions. And with that, I'd be happy to take any questions.
Okay, thanks a lot, Kevin. That was really fascinating. So questions. I have a few, so in case I'll go on. But if you have a question, Tim, yes.
What do you think the cellular mechanism that you talked about right at the end actually is? How does the how do the cells change their? How do the diatoms change their sinking speed? You said
so
maybe by increasing the radius or etc. But what's actually happening?
Right, so we're not entirely sure. There has been a lot of work showing that ion exchanging heavy and light ions could be important on sort of longer time scales. To me, it just doesn't make sense that they can do that quickly enough to do this unsteady sinking behavior. We know that actin and myosin are important. So if I had to guess, it maybe have something to do with the rearranging elements in the cell. These cells have a very large vacuole, so it could have to do with changing that sort of changing the size of that vacuole using some kind of molecular motor, but we're really not sure. We need to do the experiments to find out.
Okay, or
Yeah, hi, sorry, I don't have a camera here, so just hear me today. So very interesting. It's amazing how they can change the sinking speed, but you're assuming that they want to increase the sinking speed because that's how they gain more nutrients, but you only show the comparison to a steady sinker. I think if the scale of the patch is close to the scale of the sinking speed, then it would be much more beneficial to just go for the zero sinking, for the ambient buoyancy, because then you just stay within the patch for a longer time period, and you haven't shown how much that decreases the uptake rate. Now, it could be, as an hypothesis, that increased sinking speed is not something beneficial. It's actually an unwanted outcome of taking up nutrients, maybe because of water exchange, changing the vacuole or something, and that they have to increase temporarily the sinking speed just so they can do this water exchange or whatever other process, and then rapidly go back to zero sinking speed just to maintain their position in the patch. So as an alternative hypothesis.
Yeah, that's a really interesting point. So right, what I didn't mention, what I think you're alluding to is by sinking more quickly, they're also potentially leaving a nutrient patch more quickly. So it's sort of a balance, and it depends on the time scale, I think, of whether it's more important to increase that nutrient uptake rate or whether it's important to stay in the patch for longer. And I think it'll sort of depend a lot, and we haven't quite worked that out yet, but that's a very interesting point. Thank you.
Do you know how much you increase by sinking versus just not sinking?
So we know for the steady that so we have an equation for that based on a Peclet number, and I don't know offhand. So we have sort of for realistic values. If I can pull this back up, but sort of double to triple would be not too unreasonable.
Right.
thanks. Joe?
Hi, Kevin. That was a very cool talk. Very much enjoyed listening to it. I just had a couple of questions about the mechanics of what's going on at the cell level rather than what's causing the mechanism by which it occurs. And I'm kind of interested in the gyrotaxis that's going on, whether there's you showed us centric diatoms. So I was kind of interested in how surface areas, of course, and orientations change the factor A in the equation you showed us. And also, just to appease my mind, was there any convection in the settling chamber? Because that's a notorious problem I experience with lots of things. So I kind of doubt it, but I just wanted to check on that one. But I'm much more interested in the issue of this orientation from sort of this flipping, and we see this in lots of organisms. You see it in leaves, you see it in anything that's not axial symmetrical. It does that kind of thing and also twirls around in 3D. So I was wondering if you could comment on that.
Sure. So maybe I'll answer the other one that just sort of quickly and get it out of the way. So we find that the columns are quite stable up to maybe for about an hour. We're very careful in creating that salinity gradient. And you can kind of tell when you have a bad column, you start to see the cells, and it especially shows up in those stacks. And sort of another thing that gives it away is that the diatoms are, we know we're seeing something real because they're sinking at different speeds where we don't see kind of patches moving together. As far as the effects of sort of the morphology, geometry of the cells, that's something really interesting that we're looking into. So we have seen other shapes of cells do this behavior and sort of changing their orientations. And as you mentioned, that could affect the fluxes as well. We've seen kind of a variation too on how the orientation changes. So the Coscinodiscus cells, they have kind of a pretty fixed orientation. I think it's about 30 degrees, where they almost always sink at that same kind of orientation, which is sort of interesting. And we don't really see any sort of rocking or anything when they change their sinking speed. There are some sort of more elongate diatoms that, and I'm sort of forgetting which is which, but we have some that sort of sink on end like this, and then we have some that sink at sort of almost a random orientation. So it seems like there's something going on where different taxa are sort of have different approaches. It's quite interesting. We're sort of just getting started on that, and then you can get into chains, and then it gets even more interesting.
Thanks. Arnold?
Hey, hello. Yeah, really beautiful talk. I very much enjoyed that. I have two questions, if I may. The first one's just a clarification question. When you looked at the enhancement number and you showed that the nutrient uptake for the unsteady sinking was larger than for the steady sinking as a function of the fast speed. And I was just wondering, if you increase the V fast, do you then also lower the V slow such that the mean is still the same, that you are still comparing it to the same mean sinking velocity?
Oh, I see what you mean. So no, we treat each one independently, and we do adjust we adjust the mean that we're comparing to for the steady, but we keep the V slow fixed, and then we change the V fast. And then we have other other cases where we change the V slow and leave the V fast fixed. But you sort of, there's an argument for doing it.
So you increase the V fast, but then you also increase the mean, and you cancel it out that way.
Yep.
Okay, yeah, no, that makes sense. Okay, excellent. My second question is more, I don't know, naive. I don't know if that's actually happening or not. But I was thinking, if this diatom is actively regulating its buoyancy with, I don't know, say ion pumps on the surface, then could it also mix up its own diffusive boundary layer actively in terms of Marangoni flows or basically currents that you generate by these active processes around the cell boundary?
So I think the Reynolds numbers are too low for those kind of effects. I think, being my intuition.
Okay. Oh, yeah. Thanks very much.
Jules?
Yeah, thanks for the nice talk. I guess you may be aware that we developed in situ microscopes in my group. And I was just curious to get perhaps ideas or a community response to the crazy idea of measuring these sinking rates in situ.
I would love to do that.
Yeah, I know we probably all would. But I mean, one idea, if people put in, autonomous floats, they could measure aggregation of organisms. I guess if there's an update on anybody who's thought about doing this or would like to try it, we're in the process of trying to figure out how to develop autonomous vehicles for measuring marine snow sinking rates, which seems to be, obviously we need higher resolution and makes the problem more difficult. But I'm just curious if people have thought about this and if there are any ideas, and if anybody wants to partner on this, I would be delighted to think about it with others as well. So thank you for a nice talk.
Yeah, thank you. Yeah, I'd really be interested in seeing something like that. So as you mentioned, the resolution is an issue. They're pretty small cells, but the other thing is, and I guess this is partly addressed by using the float approach. You have the ambient motion of the fluid, and so it's a little bit tricky to account for that. So it's, you know, it would definitely be, if there's a way to do it, I'd definitely be interested in seeing that, because
Well, I can't promise anything, but I always like to think about possibilities.
yeah. Thank you.
Thank you.
Lloyd?
All right, thank you very much. It's a very intriguing talk. I'm just, maybe I have missed it, but I just wonder, how do you decide the profile when you do the unsteady, time profile when you do the unsteady sinking? Is it just something that you prescribe arbitrarily through observational experiments, or is that an optimal profile?
Sure. So yeah, I guess I forgot to mention the results I showed. We try to match the results from the experiments as closely as we could. So fortunately, we have all the numbers. We have the fast sinking and slow sinking speeds, and we have the periods of each. So we use that as a starting point, and we kind of work around those in terms of sort of realistic values for the different parameters. But we use a square wave just sort of to simplify. We could put in an actual profile of a sinking cell, but just sort of to start and to look at kind of the parameter space, we kind of started with a simpler case.
Yeah, following up on the questions, do you think there is an optimal waveform you can put in? Like, can we mathematically derive there might be an optimal time periodic sinking profile that would give the maximum nutrient uptake?
I hadn't thought of that, but if there's a way to figure that out, we don't have an analytical solution, but there, you know, there's sort of optimization approaches for CFD. So yeah, that would be interesting to see, and to see if it, you know, if it's close to what the cells are doing.
All right, thank you.
Thanks.
Thank you. Last one, Uria.
Hi, and very nice talk. I wanted to ask, first I wanted to say that there is a paper from Aleph Lug group that look at the increase of nutrient by sinking, which, and she comes to a conclusion that it's about double. And she did it with a nanosims. You can compare your result with her. This would be interesting to do. Another thing that I wanted to ask, have you ever seen the algae basically switches from sinking to going up? Because if they are always sinking, there is a, you know, on an evolutionary time scale, they will sink to extinction. So is there a solution to this? Yeah,
that's kind of an interesting, that's an interesting question. It does seem that they would, you know, you just wouldn't have any left. But and there are some cases where phytoplankton can actually sink to the surface. And there's some work by Tracy Villarreal looking at that. These we haven't seen much in the way of sort of floating, as they will occasionally float, but not often enough to really change the mean sinking speed. So
I'm talking about the individual. Have you seen one what is doing the other way, you know?
Yeah, so they will
the movies.
Very briefly, but not enough to sort of
I see.
overall. Yeah, we don't really see that, and there could be some cells that some cells do. They're a bit unusual. They're very large cells and they fast sinking. So maybe some of the smaller species.
Okay. So maybe if you put them in the dark in 4 degrees, this is for them it will signal, oh, we sink too much. Maybe it's time to go up. It's just a weird suggestion, but yeah. Okay, nice. Thank you.
Thank you.
Yeah, thanks.
Okay, let's do the very last one, Tim.
It wouldn't in the ocean, it wouldn't take much ambient turbulence to totally dominate over these very slow sinking speeds. Are you confident that there isn't much turbulence at all at the depths these organisms sink through?
Well, not necessarily. So it's sort of the two effects are they're kind of additive, right? So you have the turbulence on top of the sinking, and yeah. So we don't, we can't say that. We can't necessarily say which is more important than the other at any given time, but it will vary, I think. I don't know. I guess I don't have a better answer to that. Sorry.
To get phosphate in nitrogen, you want to go under the turbulence, under the mixing layer. So actually, you want to get sun one side. I mean, you probably know, but so probably this intermediate is quite dangerous. But you need to keep the population. So yeah.
Yeah.
Okay, so let's thank Kevin again and for the very nice talk. I'm going